Today, I will focus on how and why we use preclinical or non-clinical tests in order to find new structures or new treatment for a specific illness, and also to ensure safe use of pharmaceutical products. In this sense, we recognize that the experimentation with laboratory animals has contributed to the development of scientific area, but also in regulatory fields with a positive impact on human health. Replacement, when you talk about animal models, we refer through to three R's: replacement, reduction, and refinement. In particular, replacement refers to technology or approaches which directly replace or avoid the use of animals in experiments where they would otherwise have been used. Reductions refers to methods that minimize the number of animals used per experiment or study consistent with the scientific aims. It is essential for reduction that the studies with animals are appropriately designed and analyzed to ensure robust and reproducibility findings. Finally, refinement refers to methods that minimize the pain, suffering, distress, or lasting harm that may be experienced by research animals, and which improve their welfare, animals' welfare in particular. Refinement applies to all aspects of animal use, from their housing, handling, to the scientific procedures performed on them. As you know, pharmacology is one of the important steps of the drug discovery process. These disciplines offer strategies to study effect of drugs and how they enhance their effects, considering that there is a distinction between what a drug does and how it acts. Quantify drug activity and analyze the safety profile of new potential treatment are relevant information to evaluate. Although artificial intelligence approach could speed up and prevent failures in the drug discovery pipeline, the traditional way to study new drugs is still available and valid. Nowadays, some groups of researchers combine artificial intelligence techniques with experimental innovation to engineer a new set of processes for drug discovery. Exscientia were the first company to automate drug design and are the first to have an artificial intelligence design molecules enter in clinical trials. They focus on implementing artificial intelligence design as well as precision data generation. By focusing on both design and experiment, they are able to accelerate the preclinical drug discovery stage to enable the delivery of the new treatment of patients. But in more traditional way, pharmacology is one of the important steps of drug discovery process. This discipline offers strategies to study effect of drugs and how they enhance their effect, considering that there is a distinction between what a drug does and how it acts. In this area, pharmacology techniques also quantify drug activity and study the safety profile of the new potential treatment. We can describe three types of strategy. In vitro experiments offer more control over external variables and are less complex, making them suitable for studying specific molecular or cellular mechanisms. Ex vivo approaches maintain a higher level of biological complexity by keeping the organ or tissue structures intact. Finally, in vivo refers to research, experiments, or procedures in living organisms, such as animal models. The in vivo approach allows us to draw conclusions that cannot be obtained through in vitro or ex vivo experiments alone. Animal research is considering a key element in advance of biomedical science. Although its use is controversial and raises ethical challenges, the contribution of animal models in medicine is essential for understanding the physiopathology and novel treatment alternatives for several animals and human disease. For example, current pandemic technology such as 2019 coronavirus disease has been studied in primate, rodents, porcine models to recognize infection routes and develop therapeutic protocols. Worldwide issues such as diabetes, obesity, neurological disorders, pain, surgical techniques require study the process in different animal species before testing them on humans. But how develop the best animal model to study human disease? The screening of animals that exhibit characteristics similar to those of humans in terms of susceptibility to pathogens is essential and constitute the first step towards establishing animal models for infection disease, but also for another types of pathology. For example, expression of human viral receptors via transgenic techniques is a common method for enhancing the susceptibility of rodents to human origin viruses, which can be achieved by inserting the human receptor gene randomly or in a specific site of the mouse chromosome, and then enhances the mouse cell's susceptibility to human viruses. These methods facilitate the development of viral receptor humanized mouse model or SARS-CoV-2 in the recently pandemic. The transplantation of human stem cells or tissue into immunocompromised mice and then generation of immune of liver tissue humanized animals allow infection by strictly human tropism-related pathogens such as HIV, HBV, and HCV. Furthermore, dual humanized mice with both hepatocytes and immune cell of human origin could reproduce the HBV or HCV life cycle in vivo and also simulate liver pathogenesis process of chronic hepatitis B patients, such as inflammation, fibrosis, and ultimately cirrhosis. But also in other types of pathologies, in general more complex, with different systems involved, it could be studied this pathology with different animal models. For example, in inflammatory bowel disease, that is a recurring chronic inflammation condition of intestines, we can use different animal models to study the etiology, pathophysiology, and management of inflammatory bowel disease. Among these models, rats and mice are frequently employed due to their practically and genetic manipulability. And we use different models for different signs or symptoms that suffer the human patient. So, we can use animals for preclinical, but also for non-clinical study. What do I call non-clinical study? Study where animals are used but are applied to products that are already being marketed. They can be used for regulatory purposes or, if necessary, to solve a pharmacovigilance concern. For example, to study particular aspect of a parenteral pharmaceutical products, in particular in injections, there are several methods that can be applied to detect pyrogen. Substance pyrogen is a substance in injections that can produce fever for the presence of bacterial endotoxin. It could be described three methods, historically known as Limulus amebocytus lysate LAL test, the rabbit pyrogen test, and the monocyto activation test. But the rabbit responds for all type of agent, and Limulus test responds only for endotoxin compounds. So, to select the appropriate test or assigns, it's important to understand the difference between pyrogen and endotoxic test and what substance each method detects. Pyrogens are a group of substances that elicited a fever, and endotoxins are a member of that group. So, methods that detect pyrogen substance will also detect endotoxins, such as the study of the response in rabbits. But endotoxic specific tests will not detect the presence of non-endotoxin pyrogens. For that reason, nowadays, in medical pharmaceutical products, in different production batches, we still use rabbit in quality control in pharmaceutical research, in pharmaceutical industry, not research. Therefore, we can use animals in quality control areas of market products or in scientific or research or development areas in the pharmaceutical areas of knowledge. Thank you in advance for your attention.